Hi, I'm Stephen, and I'm a postbac research fellow at the National Institutes of Health. Scientists who study the brain typically use an animal model, like a mouse, to investigate which brain regions become active in response to certain situations, like food, stress, or a new drug. But even though a mouse brain is a lot smaller than a human brain, counting every single active brain cell or neuron in an entire mouse brain is a really long and difficult process. And as a result, many researchers simply choose to focus their experiments on just a couple of brain regions and could potentially miss important information in the rest of the brain. But for scientists who do study activation in the entire mouse brain, they typically utilize a process that's kind of like fishing, where they row their boat to every single spot in the lake in search of the fish. And as you can imagine, this would be a long and hard process. But what if we had a technology, like sonar attached to a submarine, that could scan the entire lake and tell us exactly where the fish were? Then, we could focus all of our attention on just those spots where the fish were most abundant and catch a lot more fish while saving a lot of time. In our lab, we're optimizing a technique that does exactly that, but for a mouse brain. Just like a magnetic resonance imaging machine, or MRI, can take a picture of a human brain, the tools we are optimizing create an image set from an entire mouse brain, which allow us to identify areas in the brain where the neurons were most active. But unlike a real MRI, this tool counts every single active neuron in the entire brain. In the end, we get a 3D heat map, kind of like the one you see here, where the red and orange colors mark the hotspots where the neurons were most active. Thus, researchers who use this technique are able to very quickly, easily, and efficiently identify specific brain regions that were most active in response to whatever experimental conditions they were using, which allows them to make a better informed decision about the specific brain regions they should focus on in their future experiments.